Thank you, and thank you very much for your kind introduction. So today's talk, title is Translational Medicine Informatics Approaches to Investigate Complex Diseases. So my group is focusing on the different areas like verification of the data, structuring the unstructured EHR data, synthetic data generation, integrated analysis, federated analysis, and visualization. So these approaches to close the gap between bench to bedside, to reuse the medical data for the what we call the secondary usage. um, so we develop tools to bridge the gap between uh, to medical and research worlds, and to also to ensure secure data processing. And we use these things to make predictions uh, towards the what we call clinical decision support using AML methods. And also we use the solutions using the mobile and smart sensors. And the other area is what we call from data to knowledge, to accelerate the process from data to knowledge. Here mainly to deal with the clinical and translational data integration across different data types, visual analytics, study monitoring, data integration and analysis. And the other thing is the organizing and visualization of the knowledge. So here, one area we actually working what we call the disease maps, interactive molecular mechanisms, and also we do a lot of text mining, especially structuring the unstructured EHR, EMR data to further explore. And I will touch a couple of these things in the next few minutes. So one complex issue in the drug development is the complex disease phenotypes. So how do we stratify these complex disease phenotypes? Is the reason is there are genetic features are not the same. So that is why, like, one as we know that one drug doesn't fit to all the populations. It may be effective in certain population, no effect or adverse effects in other population. Because of this reason, so this genetic stratification, or the stratifying the people into different groups, and then defining the therapies specific to that particular group. This is the what we call the personalized medicine. So to address this complex disease phenotypes, we follow a data-driven approach. I mean, the question is how to stratify this. So we follow the data-driven approach. So here we deal with a lot of clinical data coming from electronic case report forms, PROMs, PREMs, sensor mobile devices, or imaging data at the different organ levels, and peripheral markers using the mass spec or transcriptomics data from RNA-seq or in methods, and the other one is the genetics or epigenetics using the whole genome sequencing or exome sequencing. So with the next generation platforms, we have seen data explosion. So our approaches is, as I said, this is a data-driven approach. So one example is uh, uh, the Parkinson's disease study. This is what we call National Center of Excellence in Research and Parkinson's Disease. NICERBD. It is a Luxembourgish cohort, and with this cohort was built with the two main goals: early diagnosis and patient stratification, and for the discovery of novel risk factors as well as clinical progression factors of PD. So we started with the goal of with 800 cases and 800 controls with a longitudinal follow-up. So here all individuals in this cohort, all individuals goes to clinical assessment, including both neurological examinations as well as neuropsychological, like MOCA, MMCA, and so on, and followed by self-reporting questionnaires. So here we are capturing motor functions related the questionnaires and the batteries, cognition, neurosensory abilities, psychiatric symptoms, sleep patterns, and quality of life, and so on. So we have the healthy controls were followed every 4 years, and the people with Parkinsonism are followed every year. So, and also along with the detailed questionnaires, we also collect some biomaterial. Blood, urine, saliva are mandatory, and also we have optional like nasal washes, stool samples, CSF, skin biopsy, hair, postmortem brain donation, and so on. These are, and this cohort is running last 10 years, and so far we have roughly more than 5,000 total visits, to, and you can see how the cohort is evolved over the time. So it is a very rich cohort. So then, so my part was to build the complete data and data integration, data management, and computing environment. So if you look at this picture, so the study participant is coming to the hospitals, or so we also have a mobile team, because elderly patients, or they can't visit the hospitals. This van and the clinical team goes to the old age homes or other clinical sites to recruit the patients. And the other side, at the time of the recruitment, the clinical data directly goes into what we call into RedCap. RedCap is a electronic data capturing system. All the CRF are defined there, so we don't want to use any paper-based questionnaires. These are directly goes into electronic system, and at the same time, the samples collected goes into IBBL. This is a biobank. It is a certified biobank, and they have well-established protocols to process these samples, and then they prepare the aliquots, and also they document some of the what we call the sample metadata digitally. So then these aliquots goes to the different analytical labs, and we receive analytical results like genomics, we have metabolomics, and microRNA profiling, etc. And then in this on the other side also, we were using the what we call the device-based assessments. We are using the e-gate, it is the sensor devices to measure the gait patterns of the Parkinson patients, and also we have used a mobile app called Empower, and then all this clinical data, sample metadata, analytical results, sensor data, everything integrated into this data integration and analysis platform. And on the very on the right side, you see what something called SMASH, Smart Scheduling System. This we build in house, because this is meant for the clinical team to giving the appointments and so on initially, because we are not, as a researchers, we are not allowed to see the data. And also in the middle you have seen this pseudonymization. So all the data on the other side, the clinical team can see the patient name, date of birth, those things. But once the data is coming to the data and computing environment, everything is pseudonymized. Okay, so in this SMASH system, we build that one. It captures the because in Luxembourg the patients, they speak 4 languages: German, French, Luxembourgish, and Portuguese as well. So then they capture the language of preference, and also the event. And the slide is not moving.
It's fine.
Okay, and the smash system is the build for the to, and also it captures the availability of the clinicians, and also the availability of the resources. So it has some nice business logic. If you are planning 20 people in few 3 months, the system will tell you don't have resources. So this has a nice business logic, and so this is widely used by the clinical team. Today they can't run the studies without the system. So this is a nice achievement. So this is the kind of setup we have done for the Parkinson's disease study. Then other important thing, once now we have the data from the here, so this is very important to make this data, especially the FAIR. You all know what is FAIR, the making data findable, accessible, interoperable, and reusable. And in the I'm also part of the my project is finished. It's called FAIR Plus. In this one, we developed the verification process. Okay, the to make it so this we applied for this project, and mainly in a very high level, it consists of data curation, harmonization, and standardization, kind of converting the formats, data cleaning and the quality control, transformations of the data, and then making the standardization, applying the ontologies and community based ontologies and terminologies, and especially this is extremely important to facilitate the cross study analysis. Either you want to pull the data, or to analyze the to cross validation, to validate with some other cohort. And other important thing is very important for the federated analysis. You can't do federated analysis without standardizing or the interoperable data. Okay, this is quite important. So we invest quite a lot into data curation and standardization. Then the next thing is, after once you harmonize and standardize the data, we need to bring all these pieces together, what we call the data integration. So on the top layer, you can see this is the, let's say this is the data coming from the nicer PD cohort, let's say. And also on the other side, we have a lot of data available in the external databases. There is a lot of knowledge is there, to in the KEG or Uniprot, the different a lot of NCBI resources. They have DB Gap, and there is a rich knowledge is there, and the literature on the top as well. So we bringing these pieces together to the knowledge and our cohort data into systems like TransMed, I2B2 TransMed. It is I2B2 stands for Integrated Informatics from Bench to Bedside. This is maintained by a foundation maintained by Harvard Medical School. ADA is the in-house developed system. It is the old version. Currently, we are working on the new version of this. ADA is called ADAPT. It will become soon. So then, anyway, then we write what we call the ETLs. Is the extract, transform, and load. Then they load it into the systems, and they do. These solutions facilitate the integrated analysis, to, and you could nicely see the dashboards, or also they equipped with or facilitate the downstream analysis. You could do the slicing and dicing of the data and feed to the Jupyter notebook, or R Studio, and so on. So here we build the pipelines for omics analysis, and the knowledge base to support network-based analysis, modeling, and disease maps, and contextualization, and so on. And the other thing, not only analyzing the single layer, we also analyze what we call multi-layer data analysis. So here, taking for example, if you take the SNF similarity network fusion, or MOFA methods, there for the SNF, for each layer, it builds a similarity matrix, and then it fuses these matrices together, to then you will have the stratification of the samples based on the different layered information. Okay, so this is the kind of analysis we do. And this is the system I was mentioning, the ADA. So there you could bring these different layers together there, and you could organize this information into a nice tree view, because clinical variables could be quite long. They need to be organized systematically, and then uh, you can have like this what we call the dashboard functionality with the stats and views. And this dashboard is customizable for the different persona. If the clinicians would like to see certain visuals, maybe a bench scientist or a bioinformatician, a statistician would like to see different things. The tool is quite customizable. Users can have their own customizable dashboards. This is a very nice functionality. And also this tool is equipped with some machine learning approaches, to so one can do here searching of the data, doing the slicing and dicing of the thing, then loading, downloading these subsets and feed to different analysis pipelines. So this is the one tool we developed in-house. So from this Parkinson's disease study, we published a couple of publications. The SMASH I mentioned, that is out, and other one is the freezing of gait. Maybe I would like to a little bit present this analysis now. So this was mainly focusing on the freezing of gait, especially genetically stratified Parkinson's disease patients. The aim of this study is to investigate the freezing of gait uh, uh, on the cognitive subdomains. Especially how the freezing of gait is there any correlation with the cognition, in especially in the idiopathic Parkinson's disease patients. So idiopathic patients means we don't know the what is the cause of the disease. So patients without any genetics linked to the PD. Okay, in our study. So in this one, if you look at this diagram, we started with we took 571 PD patients for genotyped and which are without link it to PD mutations. So that's why these are called idiopathic patients. We don't know what is the cause of the disease. So in this group, um, 146 are freezing of gate positive and 425 are freezing gate negative. Okay, so the freezing of gate is like the PD patients when they walk and suddenly they freeze, because of this motor disease. Then out of these 571, 231 went for the neuropsychological profiling through using MOCA, MMSA, and then this is the and other part is that is where to know about the cognitive influence. That's why this set is went into this profiling, and the other side uh, other uh, other side is all are analyzed. These are the two subsets. So, and also we have certain exclusion criteria. So to remove the patients, they're going through current therapy of the deep brain stimulation or history of stroke, brain tumor, and meningitis, and those things, they will have impact on the cognition naturally. So that's why these patients are excluded from this set. And then remaining patients were their the freezing of gait has been measured, and then these two groups, one is with the patients with the neuropsychological profiling, went through comparing these two groups, mainly applied logistic regression models in this data set, both the cases, one with the freezing of gait and cognitive subdomains, that is on the right side and the left side, the whole data set after removing applying the exclusion criteria, they also went through the same analysis. Okay, so from this setup, okay, from this study design, the outcome, what we have found in this one is the patients with the freezing of gait positive individuals presented with more motor complications compared with the negative individuals, and also same individuals exhibited higher non-motor burden, including higher frequency of hallucinations, higher MDS, UPDRS. UPDRS is a Unified Parkinson Disease Rating Scale, it also has different sections. The one scores that's related to the cognition, and more pronounced autonomic dysfunction. This is measured by the self-assessed. This is the battery of questions. It's what we call SCOPA out, and at the same and also these positive individuals showed lower sleep quality along with lower quality of life. These sleep quality is measured by what we call Parkinson disease sleep scale and the PDQ-39, and these are few the questions that can give us the this information. So finally, this cognitive subdomain analysis between these freezing of gate positive and negative indicated lower scores in the Benton judgment line orientation. This is the kind of thing they have to write, and the CRAD word recognition. So all these things are, and also the higher impairment visuals, spatial, executive function, and memory encoding, and so on. All these findings are clearly indicating there is a strong correlation between the freezing of gait and the in cognition. So and also the freezing of gait we measure. Maybe sorry, I didn't mention that when we measured using the sensor devices. So more and more these sensor and mobile devices, it will make easier to find out the disease progression and in the real time. This is the use of using the sensors or the mobile devices. So then I'll move to the other part. So this is the Parkinsonism cohort PD with Parkinson's and other subtypes, and so forth, the nicer PD, and also we are also focusing on the cohorts of individuals at risk. These are not yet PD patients, but they are at risk developing neurodegenerative diseases. So we focused on the three such cohorts. One we called GBA cohort. These are the genetically stratified individuals. They have PD causing mutations, especially GBA gene, and they they're not PD patients yet. And then these patients went to functional MRI, and but they have very high chance of to developing PD. So we are very much interested to follow these GBA carriers over the time, and then to see when they converted, then we see at the molecular level what are the differences. In the similarly, other indicator what is the REM sleep behavior disorder RBD cohort. So here the people with this RBD, 80% of this RBD will develop Parkinson's disease. So it is very high signal. So that is why this is very much interesting for us to compare before and after conversion. And the other effort is a quite big effort is what we call healthy aging. There we are looking at the population level to find out the individuals at risk developing PD and or dementia in the healthy aging cohort. So RBD case to, as I said, to identify RBD affected subjects from general population, and also we are interested to estimate what is its prevalence in Luxembourg particularly, and long-term follow-up of these RBD patients, and characterize these RBD patients into high risk. We are classifying into three categories: low, medium, high risk individuals, which are going to convert into PD. So it is a very interesting to see. Later afterwards, can we build a model? To predict the certain molecular signatures. To as well. So this is a we're targeted in this one 55 to 75 years old, and the entire this age population was contacted. These are roughly 135,000 people in Luxembourg. Luxembourg population is 620,000. So then it went for the online survey, then phone interview, then mail-based screening of the BSIT. It is a small part. It is a small part of UPSIT is a 40 questions. BSIT is a quite smaller one to measure the hyposmia. And then next one is in-person visit and so on. It went for the different stages. So currently we have 56 RBD PSG proven, and seven are converted into Parkinsonism. Okay, we don't know exactly what is it. It is a Parkinson's or other subtype, and 65 are uh, um, prodromal RBD. They're going for sleep assessment, and three converted into from that sleep assessment, three were converted into Parkinsonism. So this cohort will be followed for many years to know when they're converted. That's very interesting for us. So healthy brain aging is a population-based risk cohort to identify prodromal conditions, and very similar one, but in the other case it is only focusing on RBD, but here we are focusing on many risk factors. So it's also having the online survey, then smell test. So other case we use the BSIT. Here we are using UPSIT. UPSIT is having is more elaborated hyposmia test, and then so the goal is a 300 cohort of 300 individuals, and currently we have 139 baseline visits and 46 follow-up visits. So this is the Luxembourg setup. Similarly, this is the collaboration between Germany, Austria, and Barcelona. So similar cohorts also happening in the other three sites as well. Then also then we gonna analyze all this data together, and with the goal of building models to predict the risk. So in this case, it has online survey, and for this online survey has some invite only or public. So some sites have the by invitation, some sites went for public, and then it has a website and cookie policies, surveys, and also translations in Europe. In Luxembourg, we have 4 languages. Likewise, in other countries, mostly German, and Spain has also. Yeah. So here, these questionnaires need to be translated from English to local languages, and yeah, and some spam checks and all those things. And other one is a smell test. This is the UPSIT. Then the follow up. It also has prompts and investigator assessments. I mean, it looks like small thing, but in the back end, so much is happening. You can see how many things need to be taken care to organize such a studies. And then also, like, we also build a the colleagues build a mobile app. So once you send the UPSIT to somebody, this smell test, it has 40 questions, 40 smells, and they need to give the what is that smell in that one. So instead of they sending back to us, now they use this mobile app and give the answers, and then this information directly comes into our red cap, and easily integrated into the rest of the survey data and follow-up data. So this is very instrumental. So then this slide is mainly I want to emphasize. So all the things I mentioned so far, mainly for the Parkinson's disease study, and also different at-risk cohorts. And then this is the story in 2021 when the COVID started. Okay, so then the slide you can see what I wrote is sharing the know-how. So this convened study is to understand the prevalence of SARS-CoV-2 in Luxembourg. So we have all the know-how from the NICER-PD study is directly translated into this COVID study. Maybe you could see that even the architecture-wise, we have used a lot of components. Of course, it has a additional sample collection hubs. We have 42 collection hubs in Luxembourg, and then we use a different mobile device and some other a company called TNS Ilris, they did the study. Their solution we have used to code the CRFs and so on. But at the end, majority of the components are we reuse. This is the beauty of the these systems. Once we build these things, we could reuse for many other studies. And also, like we before engaging into this kind of complex studies, we build this kind of study data flows to make it. I won't go into the details of this one, but you can see the donor is or the participant starting here, then the journey, how the information is separated. You have informed consent forms, and how the how their personal identifiable information is stored, and then then filling the questionnaires, then how then going into the sample collection site and donating their samples, and how the samples are moving to biobank. And by in this study, by evening, we need to know if there's COVID positive or negative. Then, if the positive, the clinician will contact the participant, and then it's a whole workflow. So then, because there are many stakeholders are involved, many institutes are involved, so this kind of data flow makes it very clear to who is doing what, how the samples are moving, how the data is moving, and also the GDPR is extremely important. So we prepare this kind of data flows, make it everything clear. Okay, so then I move to a bit into the analysis and the visual analytics a bit. We also develop high performance scaling the code or taking to the next level. If you take the flow cytometry, flowsum is a standard R package people use. There with that one can analyze a million cells in one time. So even with that one, sometimes it requires very high memory. So then we built something called gigasum. It is a using Julia framework. So here this was scaled into the next level. So here, by using this one, currently one can the test, this particular one is 1.2 billion cells, scaled into that level. But because using the HPC and also the parallelization, this algorithm, so this was well appreciated by the field. So this is published, and so we do this kind of what we call the hexascale computing, or building the algorithms to the next level. This is the one example. Okay, so and also now we are going taking this solution to federated setup. Okay, to if the data in the different locations, how we can analyze, not pulling the data everything into one place. So this is some ongoing activity. So other thing, also we do what we call the synthetic data, because of the data what we are dealing, dealing is everything, human data in a very sensitive in nature. But we all know that to get access to this kind of data, one need to sign the data sharing agreements, and lot of legal documents need to be signed, or the contracts, because you know when you're dealing with the sensitive data, there are certain responsibilities and accountabilities. So but to accelerate this process, to while we are signing these contracts, if I have the representative synthetic data, I can advance build the algorithms or tools and so on, to this one, or even to do kind of some uh, uh, stress test, or some testing even. That synthetic data is always helpful to because real data, when you're handling real data, there are a lot of precautions need to be taken care because of data privacy and security point of view. So this is where you start with the original data. The idea is here, and they collect certain features, aggregated features from the data, and then you build a model using the machine learning or deep learning, and then you use this model to generate the to necessary synthetic data. So the data scientists mainly provide the how many records is needed, and then some certain configurations, and then it generates the synthetic data. So we use something called synclean and cynthia to generate this synthetic data, and this is quite useful in the field. So other area I will now touch a little bit into the what we call the visual analytics, and we build certain some tools, and we are very much interested in these things. So briefly, maybe the heat map is existing since few decades, nothing novel, but we added quite some dynamics into this into this idea. You could do the clusterings and sorting out all these things happening in the real time. Okay, so this is what we call the dynamic visual analytics, and similarly, to this kind of diagrams. You could select the variables, and it's happening in the real time. Okay, so just to give an example, I won't go into the details, and this is the transmart with this the some example data from Boston Children Hospitals. So you could see like how these once you are selecting and one graph, it is automatically updating other graphs, and this is what we do this kind of dynamic visual analytics. Other area is what we call the disease maps. Disease maps is a methodology to capture exploring uh, biomedical mechanistic models happening at the molecular level. It's a very highly curated domain knowledge. We are investing since more than a decade in this area. So they will, and also we build something called Minerva, is a tool, and it uses something like Google Maps kind of API. It's not Google Maps, but it's built very similar like Google Maps. So you could zoom in, zoom out, and also another thing you could bring. These are the knowledge on the top. You could bring the molecular data as layers like genetics or microRNAs or metabolomics or proteomics data. You could overlay and see, and which parts of the map is enriched, or then you could also explain certain mechanisms. And also this is used, the model is the SPML, the systems biology markup language, and also this could be used for the modeling and the simulations as well. Okay, so and also this is the example of the recon map. Recon is the human metabolomic map. The work was done by Ines Thiele group. Then the whole this metabolomic map is coded into the Minerva. This is another also nice example. Similarly, we also did COVID map, and this is a community is formed now. There are many maps are there, more than 15 maps covering different disease areas. So other area I will briefly touch is the handling the data from the hospitals. So far I spoke about the cohorts and the organized cohorts and so on, surveys and so on. But other very important thing is the data which is in the hospitals that will not leave the hospitals because of the security reasons. And so now we need the solutions to analyze this data in the different sites. This is what, then for that one we need certain tools and solutions to analyze. A very important thing is we need to know what data is available for that one. The data, the catalogs or the registries are needed for this one, and also the FAIR is important for the data discovery. Other very important thing is the data harmonization using these like HL7, FIRE, WOMAP, CDM, and so on. And other, this is the first part, and the middle one is another challenge in the Europe. Each European country speak different languages, and most of the EHR, EMR data in the local language, like in Germany, most of the clinical EHR data in German language for us, French, German, Luxembourgish, and so on. So then translating this into English is the first thing. Then to process this unstructured data to structured data, then applying to certain standards, then data analysis. I will briefly touch these things now, and our focus especially the neurodegenerative diseases, rare diseases, infectious diseases, and immunological areas, and so on. So the one project I'm involved is called Fair Clinical. This is Sistera project. It's a collaboration between Swiss Institute of Bioinformatics and also ANR from the French colleagues, and also the colleagues from UK. So this is a project. I mean, the idea here is we start with a uh, like case reports like from the PubMed. So because that's easy to work to build the pipeline, but later the goal is to take this pipeline to apply to the unstructured EHR data. So currently we are working with the supplementary files and the case reports, clinical cases from already published data. Then mainly it has two components: the name entity recognition, and the second one is the relations extraction between these entities. And here also we are applying the LLMs and transformers, especially BioBert and CleanBert, to build to extract these entities and also the relations. And then this information is going into the what we call the structured data is OMOP. OMOP is the well used standard in Europe uh, to store the clinical data into OMOP common data model. Okay, so this is the one some activity happening in this project, and this will be taken to the goal to take into the hospitals. So other thing is the so once we have this structured data, okay, then other very important thing is that we need the federated data management and federated analysis. This is another project I'm involved, is called Clinova. This is a Luxembourgish national project, funded project. So the idea, I won't go into all the details. It has many components. We have the data integration center, and the main focus in this talk is the federated data management and analysis. This federated setup is adapted from GA4 GH compatible cloud. We don't want to reinvent the wheel, so there is some efforts happening in the Elixir compute platform as well as Elixir federated human data. So here, this J4JH compatible cloud is a framework, collection of API standards for federated data and analysis. So we are they have I mean, Elixir. I'm also part of Elixir, and so there we started with the prototype from the Elixir. Now we are taking to the next level. Okay, so the idea is very simple. The data is in the different locations, like Germany, France, Luxembourg, and so on. So one important thing is we need to know which data is available where. This is where the data catalog, collecting the metadata, the indexing into the data catalog, and the other side is the integrated analysis layer or federated analysis. So there we send the algorithms to the different sites, and they get back get back the results to the so the data will stay there, and the analysis happens at the sites and send back the aggregated results, non-sensitive aggregated results. Okay, so because of the GDPR reasons, we can't access the record level data at the patient level. Okay, so this is the idea, and so this is a very high level architecture. It has different on the top some web interface. It's kind of a one-stop shop. Then it has authentication and authorization layer. Then the next one is a catalog, data federated data catalog for the data discovery. Then other services like data repository service to deposit the data, to tell the system what data is available where. Then the second important component is the analysis component, where that has the tools registration service where the tools were registered. Then we have workflow execution service. Then workflow will have many tasks. Then we have task execution service. So these are the different layers of this architecture, and on the top and the down you have the different data uh, sites, to and then we are using Kubernetes, or even if somebody doesn't have Kubernetes, even they have simple virtual machine. Also, this infrastructure works. So the future, more and more, this is quite required to have the data in the different sites, and it could be genomics, it could be any other omics as well. And then we should be able to do the analysis, run the NVIDIA solutions like NVIDIA Flare or Flower, or even some custom developed code. This is the currently we are working on this solution. So I'm almost end of my talk. So over the years, we build a lot of tools and solutions to facilitate certain analysis. And so here, the first part is talking about this what we call fair data management. I spoke about the SMASH. This is the storing the uh, identifiable information and the clinical data to REDCap. And we are also in the process of having IRODs to having data provenance, imaging data XNAT, metadata, data catalog, mobile sensor data also we use. And then we have this data integration solutions. Some of the these are ADA is in-house. Then we are currently working on the kind of next version of this one that will be called Adapt, that will be released soon. And another solution is TransMed. We also built something called Variant DB to manage the variant data, Minerva for the all the disease maps and this graph things presented. Then all these things apply to different analysis. And other thing is data stewardship. We also invested quite into the GDPR, some tools and services to fulfill the GDPR requirements. I won't go into the details today, but it's something called DAISY, Data Information System. It is published. So this is to manage the GDPR centric metadata and to be compliant with the GDPR. And then we have something called Aspera Web and GitLab. It's common to have the code management and other things. And on the top, underneath, it's all maintained by, we have our own virtual private cloud, and we have Keyclog, identity management, and so on. It's quite, this development not happened, but it took in about 90, it took roughly now 15 years, to come to this stage. So take home message. So one thing I would say that interoperability of the data is very important. Okay, so when you're dealing with the clinical and translational data, so we have to deal with very heterogeneous data in content and format. So and also these are structured data, unstructured data in different languages. This is extremely important. It takes a lot of energy, and also like we said to the clinicians, and no paper-based clinical data collection. It is otherwise one should need to put enormous amount of effort. And other important thing is the standardization of the case report forms, and also a lot of incorporation in putting a lot of quality control checks at the data entry level. Then you will reduce the the effort you need to invest later. Data cleaning will be drastically reduced. I mean, we strongly recommend to no free text. If you add free text, you will lose its value. Yeah. Another thing, dynamic visual analytics and disease maps are very instrumental for multi-omics, or now even analysis, data analysis, interpretation at the systems level, and synthetic data, and this data privacy, security, GDPR are quite important when dealing with the sensitive human data. And and also more and more projects requiring federated data management analysis. With this one, I would like to thank my group here. It's a newly established group, and also I would like to thank BioCore from University of Luxembourg, headed by Professor Reinhard Snyder, and also I work closely with the clinical team of Professor Kruger. He's heading the neurodegenerative studies in Luxembourg, and also many collaborators and a lot of funding agencies are involved. And I would like to thank them. Without their support, we won't reach this level. Thank you. I'm happy to take any questions.
Thank you, Venkata, for sharing your expertise. It's inspiring to see how informatic approaches are being applied to tackle some of the challenges in modern medicine. So now we might answer some of the questions that are in the Q&A chat. I see there is a question. What approaches do you use to integrate image data with other modalities such as omic data?
Thanks, Mansoor. Yeah, I mean, like here for the imaging data, like we are, converting into features first, processing, anonymization, and all those things. Then going into the approaches like deep learning and the transformers, and even starting with SNF also, to those approaches, to at the end, you translate everything into a feature vector, and you look at the stratification of those things. But more and more going into the other one, the deep learning. Those are also the good approaches one can explore. Then Maria,
Question, yeah. Maria.
yeah,
What extent are
to
you able to generate synthetic data that accurately reflect ancestry differences in human population? Does the current data you have collected from Luxembourg allow you to explore such effect on disease incidence?
yeah. Thanks, Maria. I mean, this is synthetic data is to it's a degree, to what level. In Sinica, some of the Elixir involved projects, like I could say Sinica, and those are very going into quite deep by taking into the certain mutations, genetic information at the population level. So one need to build the model, here, to the endpoint. There are some efforts, and in our case, we are at the bit gross level, not at the granular level. So mainly we are preparing synthetic data for EHR data, especially clinical data. So we have some needs to share the data with some partners, and so we are at that level. But there are efforts in Sinica, and also now the IHI project called Cynthia. That is there. This will be envisioned, to even for these projects, even to IHI Cynthia, because Elixir Machine Learning Focus Group is involved there. The challenge is to get access to the representative representative data. You see, even to build the synthetic data, we need the to. So there are some efforts happening. As I said, Sinica is one, and the IHI Cynthia. So maybe it's good to follow. And I know that Finnish colleagues are quite moved into this direction. Okay, I'm happy to share the details with you. You implicit for data handling. How do you make sure they remain flexible to incorporate new data types that may be generated in the few examples, spatial omics and the other? Yes, the very important question. So these tools, especially the federated solution. Now I'm involved in the two. One project I mentioned briefly, the Clinova. Other one is IDIRA. I couldn't talk about that much. The other project. So other one, we are currently focusing on the two data types for the IDIRA. One is clinical data in OMOP and DICOM imaging data, but we are designing in such a way that the solution, we could easily plug in new data types, the spatial, the single cells, to, I mean, we are also analyzing in other contexts, single cell data in the group. So these systems we could easily plug in, to, of course, we need to develop the standards and we need to agree on the certain standard when we are going for the federated analysis. So yeah, so the systems are, you could easily bring other types. So when I discuss with the dev teams, we to have even to able to handle the, starting with the even tablet limited files to, like OMOP, CDM, DICOM or something. Yeah, to, so yeah, we are open to, because we need to prepare for that one, yeah, to handle.